So welcome to our dev session here. Give a quick introduction to myself and then NetPyNE, and then go through some demos and stuff and talk about development. I'm Joe Graham. I'm a neuroscientist, not a programmer by training. And so, yeah, if anybody has any programmer hints during my talk here, I'd love to hear them. I'm largely self-taught, and I'm not sure how good my teacher was. But yeah, I started out interested in movement and locomotion in a spinal cord injury lab. And I was looking to combine experiments and simulations because, neuroscience is very complex and you end up with a ton of data. So if you can apply it into a simulation and have it feed back into experiments, that's a beautiful little virtuous cycle, I think. We're recording from rat spinal motor neurons, and we wanted to actually model locomotion. So we wanted to simulate not just the neurons in the spinal cord, but also their connection to muscle fibers and how those pull on limbs, like in a physics engine, like a video game physics engine. Still, this is one of my long-term goals in my life here, is to make these cool biophysically realistic models with full limbs and everything. But while I was doing this, spinal cord injury affects the motor neurons in the spinal cord. They become sort of shrunken and withered. Their morphology changes quite dramatically. And we didn't have any reconstructed neurons at all, let alone real ones, or normal ones, but without a spinal cord injury. So I thought, well, I'll just grow some, surely somebody has figured out how branching patterns work, and I can just grow some neurons and then modify them to make them more like spinal cord injured ones and model with that. Turns out that's not been solved. So I ended up doing a huge amount of work growing the morphology of neurons. And it worked very well for the spinal motor neurons. Preliminary data showed it worked well for cortical ones as well. And so, yeah, I applied to the Blue Brain project and got a job there to grow all their cortical neurons. If you want to simulate an entire human brain, you need billions of neurons. And we've only got a few hundred, maybe a few thousand reconstructions at best. So I switched tracks into morphology for a long time. Then a few years ago, got back into network modeling with Bill Litton in his NeuroSim lab at SUNY Downstate. And using NetPyNE to make different network models to explore different ideas. Really enjoyed NetPyNE. It's open source, it's powerful, it does all this cool stuff. And so, yeah, for the last 2 years, I've actually actively been developing NetPyNE. And now I'm sort of stepping back to get into morphology and my spinal cord stuff. But now I know that NetPyNE's ready for me when I'm ready for my spinal cord models. So in general, NetPyNE is a Python wrapper around the neuron simulator. The neuron simulator has been around for decades now. It's probably the most commonly used simulator out there. But it's complicated, and every little thing you need in neuron, you have to program yourself. Every synapse, you have to define where, from which cell to which cell. Every compartment and every member and every model and every neuron has to be, modeled explicitly in neuron. And so what NetPyNE does is NetPyNE allows you to set a few simple specifications, and then it'll generate a complete neuron model for you, which it will then also parallel simulate it for you if you have access to high performance computing. It's very easy to have NetPyNE automatically parallelize it for you, simulate it all, and then collect all that data at the end. So it's really useful for large scale simulations. And yeah, we say it facilitates the development, parallel simulation, analysis, and optimization of biological neuronal networks. So in terms of development, I'll show the GUI next, which is a really easy, kind of fun way to start a very simple model development. But then, yeah, and you can, from NetPyNE, you can export to NeuroML, which means you can get it in any other simulator. You can also import from NeuroML. And so you can get in simulations from other places to NetPyNE, do what you want, and you can also get your simulations back out if you want to use them elsewhere. So yeah, we're trying to be integrated with the community and open source and open access and make everything work together smoothly. It's been a lot of fun doing it. So yeah, again, because it's neuron, it's you can make simulations as large as you want, as much computing power as you have. You can make a model that large. And yeah, importing and exporting. Okay, so let's do a quick. Okay, in terms of my background. You can see my personal research website at research.joegraham.name. And for all the NetPyNE website should have all the information about NetPyNE that you could need, www.netpyne.org. But right now, and you guys can follow along right now, if you go to GUI.netpyne.org and hit enter, this will automatically spool up a virtual machine for you to use developing a NetPyNE model. So let's take a couple seconds here. Yeah, and this is our, so this is the NetPyNE GUI. And it has all the tabs on top here of the stuff you need to define a model. Okay, so you got to start with cell types, and you can add a cell type, define its sections and mechanisms, create populations out of those cells, add synaptic mechanisms so they can talk to each other. You have to define connectivity rules, so which neurons connect to which neurons and how. Is it distance-based? Is it just probability-based? Is it one-to-one? Whatever you want to set. You're going to want to probably set up some stimulations, electrodes you can put in to make things happen. So you need sources and targets for those stimulations. And then you'll have to set up your simulation configuration. How long do you want it to run for? What do you want the time steps to be? What temperature do you want it to be simulated at? You know, all these different things. And then the plot settings allows you to specify a bunch of different analyses you'd like to perform on this. And again, it's very easy to build up a model. You could, if we're in the cell types tab here, click on add cell. We could add an empty cell or a basic Hodgkin-Huxley cell or a Hodgkin-Huxley cell with a ball and stick. So it's got a soma and a dendrite. Or we can also import cell templates from files. So you've got an existing cell model you want to use. You can import that into here and use it automatically. For now, I'm just going to load a tutorial so I don't have to type in all this stuff. And yeah, let's just start with the simple cell network model and take a look at it real quick. So again, I clicked on tutorials and then Tut1, simple cell network. If we look at our cells tab here, there's only one type of cell. We call it PYR. And its sections, it has a soma and a dendrite. So it's that ball and stick model. If we click on the soma, then we can see its geometry, diameter and length of 12 microns, axial resistance, all the information it needs. Topology, the dendrite has topology because it has to decide where it's going to connect to. It has to connect to a parent or it would just be floating in space. So our dendrite connects to the soma. In terms of populations, we just created one population. We called E for excitatory. If we click on that, we can see that we named it E. We only created one cell type, PYR, so it's got to be that one. And we made 40 cells in this simulation. You can set your ranges of your distributions. You can have different populations in different ranges, right? So if you're doing a cortical model, obviously layer 1 cells are different from layer 5 cells. So you can specify which densities of cells you want in different places this way. And then, yeah, to connect cells, you're going to have a synaptic mechanism. For this case, we just made one so far called EXE for excitatory. It's from a built-in neuron synapse called exp2 syn, or a 2-phase exponential synapse. The time constants for activation and reversal potential. Connectivity rules, we just have one. We only have one population, so it's our E to E connectivity rule. As in neuron, you have to set a weight from that connectivity. In this case, we're just using a generic probability of connection. Every cell has a 10% probability of connecting to any other cell. You can do this distance-based. You can do this. There's a variety of ways to define your connectivity, but for this, we've just got a very simple one. Connection delay, obviously, when a neuron spikes, it takes some time for it to travel down the axon and hit the next neuron. For this one, we're just using a fixed delay, but it's also very easy to put in a distance-based delay. So it's as if it was actually traveling in space to get to the next one. But we're just making a very simple model here. We're adding one stimulation, a current clamp, just to kick the network into gear. If we didn't put a stimulation in, nothing would happen. And we're just targeting a single cell, which should then kick the rest of the network into activity. So it goes to cell 0. You have to set the source, the target section, and the target location. And again, configuration, we're just going to run it for 200 milliseconds, a pretty short time, pretty large time step because it's a pretty simple model. yeah, in this tab in recordings, this is where you would want to set all the stuff you want to record from. NetPyNE does not record every trace or everything going on. It's an enormous amount of data. You have to specify which stuff you want to record. And this tab is where you can do all that. So you can see we're recording from cell 0. We're going to record from the soma and the dendrites. And plot settings. Yeah, we've got 2 plots set up. We're going to see the traces and the raster. So great. So let's create that network. On the upper right here, the rocket button creates the network. And we can see it in 3D. One thing I'm going to do real quick is I prefer the black background. So I think it makes it much easier to see what's going on. In the control panel here, we can randomize the colors just so it's easier to see which each one is. And yeah, so that's our network layout in space. But it's just, we've just created it. We haven't run it yet. So if we want to run it, we want to click this rocket button, simulate the network. And that'll actually call Neuron, run all the simulations. And it says I've already simulated. Great. So now we should be able to look at the analyses. On the left-hand side here, we can see there's a connections plot, which there's only one population here. So it's not very interesting. There's our excitatory population and our current clamps with different strengths. Traces will be more interesting. So this is cell 0 in population E, the dendrite and the soma. And you can see that we put that stimulus into this cell at a time of 20 milliseconds. And you can see we put it into the dendrite. So the dendrite voltage actually increases there after the stimulation, which drags up the somatic voltage until we get that first spike. And then we start seeing regular spiking in this cell. To see it, in the entire network, we can go to the raster plot, which is just on the bottom axis here is time, and this is the cells of the population on the left here, and this is how they spike over time. And I should be able to zoom out a little bit. Yeah, zoom out a little bit. And yeah, you can see that nothing happens until cell 0. It gets a spike at 24 seconds. It gets input at 0. It takes a while to increase the dendritic voltage, hit the soma voltage, it spikes, and then that transmits to the cells it's connected to, which transmits through the network and sort of kicks it into gear. Cell traces, that's the only one we have. Yeah, there's a variety of different analyses we can run. So a spike histogram, for example, it's not really a histogram, it's average cell firing rate over time, but obviously it's 0 and then gets some oscillations in the frequency there. So yeah, this is just a very simple model to show you that if you're going to develop, start out with small, simple models, the GUI is an easy sort of fun way to do it. And then, oh yeah, one other thing with the GUI that's important is that it's got its own Python terminal here. It's like your own little Jupyter notebook in the GUI. So anything I change here will automatically be reflected in the model. Anything I change in the model will automatically be reflected in this Python. So you can't do everything you might want to do in the GUI. We're getting there, but it takes time. But you can do anything you want in NetPyNE in the GUI by doing it programmatically in this little Python box. Importantly, importing and exporting. So let's just export this little model to Python, and then we can use it programmatically in Neuron. Yeah, I'm just going to call it output. Export it. That gives me a different name, but that's fine. And I'm just going to save it on my desktop. And yeah, let's take a quick look at our model here. Yeah, you got to import specs and sim from NetPyNE. You have to create 2 objects, your network parameters, then your simulation configuration. And yeah, here we can see all the definitions. It's a very short number of definitions we need to create a fairly complex model. So we got 40 cells of the pyramidal type. Here we define that pyramidal type. It's got sections. The soma has a geometry and a mechanisms. The dendrite has geometry and its mechanisms, as well as its connectivity to the soma. Define our excitatory synapse model, set its properties. And yeah, here's our connectivity. So the preconditions are the presynaptic population, the postconditions are the postsynaptic population or set of cells or whatever it is. You can define this in many different ways. But for this one, yeah, just E to E and a probability of 0.1. And so, yeah, so we have the seeds set, the random seeds set to be constant. So if you run this over and over again, you'll get the exact same output, despite the fact that it's random. If you want to see the true randomness, you'll have to go into the sim config and set a different random seed. And then, yeah, it'll make a different new network based on these properties every time. And that's a nice way to compare robustness of a model, for example. Run it randomly a bunch of different times and see if it still behaves the way you'd expect. Stimulation, source, and target parameters. Again, we just did a current clamp into that one cell to kick it into gear. And we ran it for 200 milliseconds, recorded from a few cells, and then, yeah, specified a few plots to be generated automatically. So, yeah, so like I said, the GUI is a nice, fun way to get started on a model. But then as you get more complex, right now, there's no way to run the GUI yourself. It runs on, I believe it's open source Brains platform right now, and they only give you one core, when you're doing this. So for large, big models, you're probably going to want to go to the programmatic instead of the GUI. But yeah, okay, so that was our GUI demo. So now, yeah, let's do a programmatic demo. We'll do the same thing programmatically. I'm going to close the GUI, and great. Yeah, so I'm going to show you this is actually how I would go about making a change in NetPyNE right now. I'm going to open a new terminal, and I am going to change to my desktop, which is where I do most of my work. And I'm just going to make a dir called devsesh and change into that. Okay, so I'm in this directory on my desktop now. I don't have NetPyNE installed on my system all the time. It's constantly being updated. Its dependencies are constantly being updated. So what I have is I actually have a few aliases or functions in my bash RC or my ZSHRC file that automatically set up my NetPyNE environment. So I call these venv. So for virtual environments, I've got venv NetPyNE and venv NetPyNE dev, which set these up. NetPyNE dev pulls from the development version. NetPyNE itself uses the latest pip release, to see what actually is out there right now. But yeah, so if I do venv NetPyNE dev, that is going to create a whole virtual environment for me, install NetPyNE, install all its dependencies, and do all that. So quickly, while it does that, I'm just going to show you my ZSHRC file. So this is in your home directory. Typically, if you use bash, it'll be .bash rc. I use ZSH, so it's ZSHRC. But if you open that, ZSH has a bunch of default settings. And then down here, I've got my venv NetPyNE dev. So it prepares a virtual environment for NetPyNE using your system install of Python 3. Creates a virtual environment. This is built into Python 3, so you don't have to install anything for that. Then you activate your virtual environment using this source command. And then you want to install and upgrade all the stuff that it needs. So install and upgrade pip before you do anything else. Wheel, IPython. There was a problem with a version of Neuron not too long ago, which required this extra step to install a couple things specifically, but that is probably fixed by now. Install and upgrade Neuron. And then, yeah, we clone NetPyNE, and it's the dev branch is the default branch. So you get the dev branch. And then we use pip install -e to install it locally. And then I also install an IPy kernel and a Jupyter notebook, so I can immediately open any of this in a Jupyter notebook, which is often handy, especially for sharing things with other people. And creates a kernel for the Jupyter notebook. And then, yeah, it's ready to use. And so, yeah, this went through all that. And you can see I've got env on my terminal here on the left. So I know that I am in my virtual environment. So I'm going to close. And any questions or comments at any time, please jump in. Let's see here. Right now, I want to go into yeah, dev session. And yeah, I'm just going to drag this, what we exported from the GUI into this dev session folder so I know where it's at. Okay, so yeah, and then we should be able to IPython -i NetPyNE _init, that long file name. And this should do the exact same thing programmatically without the call to that, to the GUI. Desktop dev sesh. And it might take a while to spool up the first time here. Ooh, oh, well, it didn't seem to do anything. Let me just look at this file real quick. From NetPyNE import, create things. Yeah, when the GUI outputs it, the GUI wants to run this last line itself every time you click simulate. So you have to uncomment this last line in the output if you want to have it run automatically. So I'm going to save that. Cancel. That's yeah, save as dev says, save as that. Replace. Okay. And so now let's try that again in IPython here. IPython-i, and then run that file. There we go. So now we can see created a network of one cell population on one host, 40 cells, went through the simulation, the analysis, and plotted. And okay, it didn't actually save those figures. Because we don't have save fig equal to true. Let's do this one more time. I'm going to change show fig to save fig and save that again. Exit. Run that one more time. Okay. Oh, well, yes. And this is plotting the interactive plots. By default, the GUI is going to use the interactive plots instead of the static plots. But the interactive plots are fun too. You know, you can turn off either any trace you want. When you zoom over it, you know, you can see what the actual values are. Let's look at here. Okay. Yeah, so we've got, let's get rid of iPlot traces and iPlot raster. It'll, save fig. True. Save fig. True. Okay, save that. Okay, so that should get rid of the interactive plotting, and I'll just close this and go back and run that one more time. Great. Okay, well,
can I ask a question?
The raster plotting, yeah, anytime.
Yeah. How often do you find yourself using the GUI versus like scripting and the traditional command line? Like, do you use either one for different use cases or different parts of the workflow?
I mean, typically the GUI is really useful for people who are just starting to get into this. It's faster and easier for me to do it programmatically at this point. Okay. I mean, that's not entirely true. When I'm starting a brand new one, so a while back I started a spinal cord model with a few other people just for fun, really, and we used the GUI to develop it just because it's nice to see it right away and to be able to move it around and play with it. But we wanted, you know, hundreds and hundreds of motor neurons, and the GUI was a little too slow for that. So we just did like, you know, 10 of each population and got it laid out and everything the way we wanted it, and then switched to programmatically for the more, you know, memory intensive simulations and stuff.
Okay, thanks.
Yeah. Okay, so yeah, so anyways, that's this is how I develop NetPyNE. I create a new virtual environment. Now I can open a new tab here, and I'm going to change into NetPyNE. Now I can see it's in Git. So I can Git status. I haven't changed anything, But if I were to modify a file now, it's just in this little location. I can work on other projects and other bug fixes elsewhere in different virtual environments. And when I'm done, I can just commit this and push it to Git and just delete this directory altogether, which is, my workflow typically. Keeps it nice and clean, I find. Okay, so that was, the GUI and into the programmatic. Okay, so now I was just going to talk briefly about sort of, I made a ton of bug fixes and a ton of small improvements, but I did sort of two big things while I was at NetPyNE there. One was documentation. And we wanted this to be really well documented. We wanted it to be very easy to use for users and developers. And so that was a big push when I first joined NetPyNE, was to do the documentation. And specifically, what I did is, I think all the links are on here. So we use, we document code inline and online using doc strings, Sphinx, Napoleon, and AutoDocSum. So you can see these links here. Doc strings are just the strings, three parentheses or three quotes on either end of it, and it's a big long block quote. And anytime that's right after a function or a module or anything in NetPyNE, that's going to be a doc string. So I'm just going to look at our GitHub real quick and go to one that I know has a good doc string. I'm going to look at plot raster. So here we're looking at the the code itself. But right at the bottom of the definition, it starts with these three quotes, and this is a doc string. This is a function to produce a raster plot of cell spiking. It's got a bunch of different options you can have, various parameters, and different plot options, whether to show it, whether to save it, overwrite it, all that stuff. And this is nicely human readable. So this is not in RST, which is what Sphinx uses RST, restructured text, or rich text, whatever it is, to produce nicely formatted HTML documentation out of this. But it has to be RST, which makes it very hard to read. So Sphinx is what takes any RST files you have and makes those into HTML or PDF or LaTeX or whatever you want. It's a very useful program. But again, it can only read RST and translate that. And so to make ours, our documentation readable both in the code and online very easily, we combine Sphinx with Napoleon. Okay. And what Napoleon allows you to do is make human readable doc strings. So normally, if you wanted to make nicely formatted HTML doc strings, you would have to include all this RST formatting. But I don't like that. It makes it hard to read. I'd rather much read it human readable, either in Google style or NumPy style. We actually chose NumPy style instead of Google style. But it allows you to write this in a human readable way, but it still gets nicely formatted into your HTML documentation. So Sphinx, Napoleon, okay. And then AutoDocSum is a Sphinx add-in or plugin that allows you to create automatic summary tables of all your documentation. So let me show you what that means here. Let's go to netpyne.org. So our package index is where all our HTML documentation lives, and this is all pulled automatically from those doc strings in the code itself. And so what AutoDocSum does is creates a nice table of contents. So these are all the sub packages in NetPyNE. We've got analysis, batch, cell, conversion, metadata. Let's look at plotting. So I know it's nicely documented. If we look at netpyne.plotting, we can see plot LFP locations, LFP-PSD, all these different functions that are in there. Let's click in further all the way into plot raster. And now it's got this nicely formatted documentation that shows you what all your options are, the default values and all that. And so this is, again, it's fairly simple to accomplish with Sphinx, Napoleon, and AutoDocSum. right now I'm going to go into, my dev session, my virtual environment I just made, going into the NetPyNE directory, and then doc is where all our documentation lives. Everything lives in this source directory. Our index, how to install, our reference, our tutorial. These are the RST files that get converted into HTML for the website. And so build.py. Okay, well, first of all, I guess, okay, conf.py, config.py in your source code. When you set up Sphinx for the first time, it'll make you a default sort of setup like this, and it'll have this conf.py, config.py file. And it'll be fairly standard, except you'll want to add the extensions you want. So we want to use Napoleon and AutoDocSum, for example, so you have to make sure those are included there. For Napoleon, they only have a few set of section names, parameters, arguments. We wanted to have a few unique ones, so you have to add those here. We want to call them NetPyNE options. We'll call them plot options. We order it by source in the file instead of alphabetically, because usually our most important classes are at the top. And yes, so this HTML theme is Sphinx. Is there anything else important in here? I don't think so. Yeah, so once you get your config.py file set up with that stuff in it, then in Neuron or NetPyNE, we use this build.py Python script to do all this. And yeah, this is sort of nicely documented, tells you everything you need to do. In order to build the documentation, you first have to install Sphinx and its theme and AutoDocSum. And if you want to put it up to pip in those, you'll have to install Wheel and Twine. So I'm just going to go to my guy here, exit out of IPython. I'm still in my env, my virtual environment. So I just paste all those installs, it'll install Sphinx and AutoDocSum and do all that. And then, yeah, what this file does is if you have a previous build directory, it deletes that to avoid any problems. It keeps these RST files that are standard in our thing, that aren't generated by Napoleon. Then it deletes all the ones that are not that. So it creates an RST file for each, plot raster has an RST file, which makes that nice HTML documentation. We delete all those just so there's no problem with overwriting or anything. And then, yeah, you use this Sphinx API doc to generate all that documentation. And then we actually rename a file. We don't want it called, we want it called package index instead of NetPyNE or whatever. So quick, fix the heading of a file real quick. And then, yeah, Sphinx build, source.build is what creates HTML out of all those RST files that originally come from your doc strings. Yeah, this is something I'm pretty proud of, to have it nicely. Documentation is a pain because you've got it in the code and online. You got to keep both updated. This is, to me, it was a nice clean way to only maintain it in the code and then have it automatically built into nice HTML documentation. So yeah, that was documentation. And at this point, yeah, my other big project was trying to separate analysis and plotting. Yeah, separating analysis and plotting, separating and standardizing. NetPyNE evolved, as many software projects do, from Salvador, the PI, his work. He needed to build large scale simulations using Neuron, and he wanted to be able to do it simply and easily and repeatedly. And so, yeah, so he made this sort of Python wrapper, which became NetPyNE, to wrap around Neuron and make that all very simple. Problem is, when code evolves over time, you don't necessarily think in advance about modularity and all that. And so, yeah, the analysis and plots sort of evolve together. You know what I mean? It's all like, there's no separate analysis routine and then a plotting routine. It's all analysis and plotting in one, and the output is just a plot. And we wanted to be more modular than that. And we wanted our plotting to be more powerful than that. We want you to be able to plot into any axis, right? Don't just create a plot every time you want a raster. What if you want to overlay a raster on something else? What if you want to So we wanted it very modular, separate the analysis and the plotting. And yeah, a few advantages. Other advantages of this is it allows anybody to use these plots. So even if you don't use NetPyNE for simulations or anything else, I think our plots are pretty good. And this allows other people to use those with their own data without running it through NetPyNE first or anything, which I like. And I wish other people would also make their plots useful in this way, so I could use awesome other plots that people have developed.
Joe, sorry, I was just going to ask you, but even though all of this functionality is not necessarily NetPyNE specific, does it still live within the NetPyNE package, or is it a plan to split it out into its own like analysis and plotting?
Good question. No, it all still lives in the NetPyNE ecosystem. For example, now when I got to NetPyNE, we did not have plotting / plot raster. We had analysis / plot raster. Okay, and that did the plot raster analysis and the plotting and spit out the figure. Now it's separated into plotting plot raster and analysis prepare raster, which is in spikes. So yeah, here we've got prepare spike data, prepare raster. So all our analysis functions now begin with prepare. And those spit out a little dictionary of exactly all the data you need to make your plot. So this ensures that we're exporting all the data necessary for the plot, and that if you have all that necessary data, you'll be able to make the plot. But yeah, so for example, let's just try this real quick. I haven't run this in a while. I hope there's no bugs. But we're going to do that IPython. We're going to run that simulation we exported. And now we're going to say raster data equals sim dot analysis dot prepare tab. Let's go prepare raster. Okay, and then let's print raster data. Okay, let's just see raster data.keys. Raster data.keys. There we go. So yeah, prepare raster spit out a dictionary with the spike times, the spike indices, the spike GIDs, the number of cells in each population, the labels of those populations, if there's any net stims, which to include the time range. All the information you need to be able to reproduce this plot is in this dictionary. So anybody can take all this information, plot it elsewhere, do whatever they want with it. But also, if you get your data into this format, which is a very easy format, then you can use, then you can plot it using the plot raster. Sim.plotting.plot raster data equals. Again, I haven't tried this in a while. It might be slightly different. Okay, data is not the keyword I'm looking for. This is spikes. We want plot raster. Raster data it needs to be. Okay. So, although this should be changed to just data so it's consistent across plots and you don't have to remember the name. But yeah, anyways, so if this it just took that data I just output and used it to plot it. Show fib equals true. So this is not, it did not run the analysis, did not get this out of NetPyNE. It just used that dictionary we just had to create this plot. Allows, make a nice clean separation between data and plotting. So you always export all the data you need, and your plotting can always accept outside data to do this. It makes it more modular, maintainable, and sustainable. And so as part of this analysis and plotting improvements, I haven't done much object-oriented programming in my past, but this seemed like a perfect opportunity, a really good time to do it. A MATLAB or a Matplotlib figure is an object with objects inside it. Could have multiple axes, it could have data, it could have all this stuff. So we created a meta figure object that holds all this stuff. And it also holds all of our methods and functions that we can use on a figure, saving the figure, showing the figure, saving the data, all this stuff now lives in one nice clean object we call metafig. And yeah, let me just pull up metafig here at some plotting plotter. And oh, I didn't redo, the ordering is important. See, this is alphabetical, but it should be, the meta figure should be at the top since it's the most important. I have fixed this. I just haven't redone the HTML since. But yeah, so if you look at this meta figure class, it's essentially, you know, an object that can hold a matplotlib figure. And it's got a variety of things you can do to it, you know, that you want to do to every figure. Do you want to add a super title? Finishing the fig, we've got a few set things we do. Saving the fig, showing the fig, these all live in one place. And then a meta figure can hold multiple axes. You know, you could have multiple subplots and whatnot. Each one of those axes is going to get its own plotter, which are all based on the general plotter class. And again, this is just allows us to sort of put all the stuff we do to figures in one nice, clean, modular place. So whenever we modify, add color bar, it affects all our figures, you know, color bars, legends, scale bars, formatting the axis, loading or saving data. These are all things you do on individual axes. And yeah, and then each general plotter can be a more specific type of plotter to make it easier to do a histogram or an image or a line, or you can just plot directly into those. And yeah, documentation, analysis, and plotting. And yeah, I think that's about all I have. So yeah, I'd love to hear comments and critiques and anything now or questions.
Yeah, I asked because it almost feels like all simulator developers have their own analysis and plotting routines, because we have the same thing in NeuroML, right? You have a class with plots, rasters, and all that. And at some point, I was wondering if it'll make sense for all of us to try and agree on. I mean, now there's agreement. There's a standard for experimental data, and I know this has been discussed before. I think before you and I got into simulation development to try and standardize simulation output, right? Some of it was done in Sonata, for example. So the ideal scenario would be that we all save all our simulation generated data in a standard format, perhaps as close to NWB as possible, so that it's also very similar to experimental data.
Yeah, I agree.
Right, and then that way we don't each have to develop our own analysis and plotting routines. We can just reuse what the massive NWB ecosystem is already developing on. I mean, they're doing Python, they're doing MATLAB, they're doing R, they're doing, right, and then you can make all of these fancy. But I think that might require a bit of coordination between our working group and reaching out to people. And
sure, I think it's a worthwhile longer term goal. I think that would be great if we could get to a more sort of standardized data format, so tools play together more nicely and stuff. And yeah, NWB, you're right, seems to be like maybe what we should build it around. People might have to add different things or whatever, modify it slightly. But yeah, I think working towards an NWB format like that is a good idea, and I'll talk about it in our next NetPyNE meeting.
Yeah, because we started looking at, but it's not trivial to just output your simulation data in NWB, but it is quite easy to convert, for example, a raster spike file into an NWB file, right? Because they already have fields for spike times and spike trains and all of that. And NWB, because it's done for experiments, everything we model we model experiments, right? So everything we store corresponds to an experiment in some way, whether it's membrane potential or if it's calcium imaging, whatever, blah, blah, blah. So it should be doable. It's just that I think we're also used to doing our ad hoc post-processing and ad hoc plotting that we haven't gone towards an NWB compatible simulation data format yet. But yeah, I mean, that would be quite useful. We could discuss it in one of our working group meetings.
Yeah, I think that'd be a great idea. Let's add that to our next agenda, actually.
Yeah, as a start, yeah.
James, you got your hand up there?
Yeah, hey Joe, that's a really nice talk, first of all.
Thank you.
So I was going to say there is a standard called NEO, which aims to cross the experimental computational boundary.
Okay.
Like Pine uses it, but the problem is that honestly, it's not very nice. It's kind of annoying. So I fear a lot of people, because it's annoying, just stick with ad hoc stuff and convert when necessary.
Right.
I'll try and find a link to it.
Yeah, if you could share a link, that'd be
yes,
great to have as a discussion point. Yeah.
As Anki can beat me to it, which is quite possible.
The good thing about Neo is it does provide a way to load and export to NWB as well. So
that's very useful.
It's like conceptually, it's very nice, but the problem is that handling the data in the Neo format is much more annoying than
in practice. It's a little.
So Joe, a really nice talk. I'm really glad that we have it recorded, because in 40 minutes, you've given us a nice overview of NetPyNE. So I do have a few questions for you, but more at the general level. So you mentioned that NetPyNE is like a Python wrapper for Neuron Simulator itself. So I presume for the GUI app, there certainly might be several, I presume certain limitations that can't be done what you could do on Neuron. But for NetPyNE programmatic, is that true? Is there some aspects of the Neuron Simulator that can't be reached?
No. Ultimately, anything you can do in Neuron, you can also do in NetPyNE. It just might get a little complicated. Most of the stuff people do commonly day to day, we've already made it very easily and nicely integrated with NetPyNE. But if, for example, if you're random example, controlling a robot arm, every time step you got to send out or receive information, you know what I mean? Neuron can do that, but NetPyNE isn't set up to handle it automatically. You have to go in and handle some of that yourself. But yeah, ultimately anything that can be done in Neuron can be done in NetPyNE. Most simple Neuron stuff is already very easy to do in NetPyNE. But the GUI is even more limited. The GUI does not do like a bunch of the reaction diffusion stuff right now, for example.
So as an example, for gap junction models are already doable in NetPyNE?
Yes, people have done gap junction models in NetPyNE. I believe there's an example in our GitHub.
All right.
But yes, it's possible, and I know it's been done.
All right. Okay. Yeah, because in Neuron, the gap junction implementation is let's say a bit unusual or some. You either sort of specify the resistive pathway in between, or use the linear mechanism for specifying the equations itself.
Yeah.
So okay, so I'll check that example out. A related question. So NetPyNE, you did show that NetPyNE you could export to a Python file, the source code, but I guess that particular file that you showed us was NetPyNE specific.
Right. Yeah, we just exported to Python. Yeah, from NetPyNE. To export to NeuroML is slightly different.
All right.
Where's the GUI? I think you can do it from the GUI, but I'm not sure. You can definitely do it programmatically. Obviously.
Yeah, I was just wondering if there was a way to sort of create this nice model using the GUI app and then have it exported. And if somebody wants to use it on Neuron Simulator itself, is that doable?
You mean without the NetPyNE wrapper?
That's right. Yeah.
I'm not sure we have a way.
So as a use case, what I'm perceiving is, for example, if you want to run it on Neuroscience Gateway, I'm not sure if NetPyNE's already there as one of the simulators.
We should be. We should. We've got a nice collaboration with Neuroscience.
Oh, all right. Okay. I haven't checked it in a while. Okay, fine.
Yeah, it's fairly new.
Right.
But yeah, so no, you should be able to do it there anyway. Yeah, it looks like the GUI does not automatically export to NeuroML or anything, but certainly the code itself does.
Can I expand on that question and sort of ask a very similar question, which is like, what is the coverage like for the NeuroML export? Is it like pretty much any valid neuro or any model that like successfully maps to NeuroML is going to work, or is it still kind of like a little bit untested and like only simple models for right now?
I haven't actually done any NeuroML myself. I just work in pure NetPyNE. I've never had a reason to export or import yet, so I haven't done much myself. Poreg Gleason is the real NeuroML hero who does a lot of the work for NetPyNE NeuroML stuff. I believe it's pretty solid. It's not just simple stuff. I believe anything that can be done in NeuralML can be exported from NetPyNE now, but
Awesome.
don't quote me on that. There might be some bugs or something still.
Thanks.
You did briefly mention that NetPyNE is constantly evolving, lots of updates, partly the reason why you reinstall it afresh when needed. So how stable would you say it is? For example, if I start developing a model on NetPyNE right now, 6 months down the line, would the latest version be backward compatible?
Yes, we make a huge effort to make everything backwards compatible.
That's great. Okay.
So yeah, even we still have the original combined analysis and plotting in there to maintain backwards compatibility. So now we would use sim.plotting.plotraster, but if you use sim.analysis.plotraster, it uses the old code and everything. So it's absolutely backwards compatible.
Okay, that's great.
Yeah, we don't want to break things constantly. We want to.
Yeah, that really puts developers off. So one last question for you, but that might be taking a slight detour. So sometime back, I heard about this middleware called Geppetto, and I believe NetPyNE uses it. And I've always been curious, what does Geppetto exactly offer, and how does it sort of plug into NetPyNE?
Okay, so we work with actually a private company called Metacell for the GUI.
All right.
They're great. They're all neuroscientists. And they developed Geppetto. Again, I'm not a programmer, so I don't really know much about Geppetto and all this. Geppetto, I know, is like their underlying web code to make this happen in the GUI. And I believe that's open source and everything, so they're making that available to others.
All right.
They use it themselves to do this GUI stuff. Yeah, contact Metacell if you have more questions about that. I really don't know too much about it.
Sure, but that's restricted to just the GUI app, right? Not NetPyNE in whole. Okay, that's great.
I can actually shed some light on that because the Geppetto frameworks also used in open source Brain version 1.
Okay.
All right.
Quite an old framework, and it's also used in NWB Explorer. So the idea is that in order to build a front end, you need a sort of standard model, right, which will then map onto front end elements. And what Geppetto does is they have their own meta model, and they convert whatever needs to be converted into this meta model so that all their standard UI elements can then just work with that meta model. So for example, whatever your NetPyNE is doing, a dendrite or whatever, it will get converted to a corresponding entity in the Geppetto meta model. And then they only build UI elements that can render this Geppetto meta model, right? So for example, a raster plot will correspond to a particular thing in the Geppetto meta model. And so NetPyNE just has to generate it. You have to convert it into a Geppetto meta model, and then it can be rendered just based on the standard UI elements. And so NWB Explorer works in the same way. You take an NWB file, and there's a mapping from the NWB standard to this Geppetto meta model. And then all the elements that are, for example, a lot of the plotting that's being used in NetPyNE UI is also being used in NWB Explorer, because all the front-end elements need it to be represented in this Geppetto meta model, right? So it's just an abstraction layer that allows them to build front ends. Right.
All right. Okay.
It's evolving. So open source Brain version 1 had Geppetto in Java. Everything is being redone in Python now. So for example, NetPyNE UI uses Geppetto meta, which is completely in Python. I think the new frameworks are Python, and they're using React for rendering and everything. So open source Brain version 2 is in React. NetPyNE UI is in React. So they've really done a lot of it to make it more server-side and lightweight.
All right. Okay. Yeah. Okay. Thanks.
Yeah, if you guys need any GUI development done, I would highly recommend Metacell. They do great work and they're fun to work with. And Eugenia.
Yes, hi. I wanted to ask about caching. Have you considered implementing automatic caching in NetPyNE for simulations? And would it be useful for development?
So again, I'm not a programmer. By caching, you mean like sort of saving some data as you run it?
Yes, saving the parameters of the network and then memorizing the results. And then every time you run the same network with the same parameters, you get the results automatically loaded.
Oh, that's interesting. I'm not sure we've really talked about that much. And we do have a couple, it's not really caching, but we do have ways to save data more.
Yes,
if you're running a massive simulation across thousands of processors, you can save the data from each node in each node, and then at the end collect that together and put it back together. We also have a what do we call it? Interval save, where if you're running a really long simulation that might crash before it finishes and you lose everything, it'll save, every so often that you want. So it's not really caching, I guess, but.
Yeah, I saw that you can do this manually, but it's not automatic. And I wondered whether, like, during the development, would it be good if it was automatic?
No, that would be a cool feature. Yeah, potentially quite useful moving forward, but no one's done it yet. Join us, help us do it.
Okay, cool. We've hit the 1 hour mark. Let's stay another 5 minutes in case we have any more questions. Otherwise, we'll end the meeting. Joe, that was very, very interesting, like all our dev sessions are. Even though we all use strings and everything, there's always some new bit of information you pick up when somebody explains how they've done it.
Yeah, these dev talks are great because, again, I haven't been exposed to a lot of the way people do things. So just seeing other people's workflows has been immensely useful for me because I would have no idea. Oh, you can do that, huh?
It's really nice, yeah. So
I find these sessions very useful.
We'll try and do one on NeuroML soon. I have a ticket open for that. Yeah, but NeuroML is Java and Python and all sorts of things in XML. So I think that might be a bit heavy, but we'll try and do one on there.
Yeah.
To all the people here, if there's a particular tool you use and you'd like us to try and organize a dev session, let us know, and we will reach out to developers and ask them to do it for you, right? And of course, I know the impression people have of the software working group is that it's all just a bunch of devs, but that's not true. So if you are just interested in general in all the software you use for your work, please join the software working group, right? We want to build a community, and we want to be able to exchange knowledge and information and ideas. And we just want to hang out really with people who have common interests. That's the idea of the working group. So I'll share the link to our website. Yeah, please join us. It's completely free. You do not have to be a member of the INCF or OCNS. And all our work happens in the open on GitHub. Issues. We don't even have a mailing list, right? You just join us, we add you to a GitHub team, and that's it. And then you're just there with us. All right. Okay, cool. Yeah, another 2 minutes, and then I think we'll close, and everybody can get on with whatever they were doing before this joined.
Get back to our work lives here.